Good morning. Thank you for joining PPMD's webinar today with Capricor regarding their HOPE trial of CAP-1002. I'm Kathy Kennett, Senior Vice President of Clinical Care with Project Muscular Dystrophy. We're excited to be joined by John Lynn Jeffries, cardiologist and PI from Cincinnati Children's Hospital Medical Center, Deborah Ashheim and Linda Marvan from Capricor, as well as Kath Diashura from Coalition Duchenne. Just a few housekeeping points before we get started. First, you'll note that the audience is on mute. If you have questions, please type them in the chat box in the lower left corner of the screen. Many of you have submitted questions for our speakers. We'll try to get to those questions after the presentation, but I may interject questions if needed during the presentation. Also, a copy of this webinar will be archived on PPMD's website for viewing later. I'd like to now introduce my co-moderator, Kath Diashura, Executive Director of Coalition Duchenne. Kath?
Hello, good morning, everyone. Thank you so much, everyone, for being on the call today, and thank you for Parent Project Muscular Dystrophy for organizing this webinar. So my name is Kath. Most importantly, I'm the mom of Dusty Brandon, who has Duchenne, and he is now 24 years old. I would just quickly like to provide a little bit of background before we get started and before I hand it over to the Capricor team. This work is something very close to my heart. At the end of 2011, I read an article called Repairing Broken Hearts in The Economist magazine. Among others, it featured Dr. Eduardo Marban at Cedars-Sinai and his work with cardiospheres for heart attack scarring. I sensed the potential for applying this rapidly evolving treatment to Duchenne, and it became sort of my quest to see cardiac stem cells applied to Duchenne hearts. I wrote to all of the leading researchers mentioned in that article, and only Dr. Marban responded. So what began was collaboration with him on the idea that scarring in Duchenne hearts may respond in the same positive way as the scarring with heart attacks. We funded the initial small mammal studies and other work in his lab, but this concept has now been realized into a potential treatment for Duchenne hearts. It's simply incredible. We're excited that Capricor Therapeutics chose to commercialize this work and embark upon the HOPE trial. I'd like to thank everyone at Capricor Therapeutics, John Jeffries, of course, and the 25 boys who participated in the trial. So I'd now like to hand it over to, I guess Kathy will introduce, I'll introduce Linda Marban, President and CEO of Capricor Therapeutics. There's Deborah Ashheim, Chief Medical Officer of Capricor Therapeutics, and John Jeffries, the Principal Investigator of the HOPE trial, to discuss the 6-month results.
Thank you very much, Kath, and thank you, all of you, for joining us this morning. We are delighted to be here, and it is truly due to the tremendous energy of Kath and then the Parent Project for Muscular Dystrophy, and specifically Pat Furlong, for helping us get to this day. So without any further ado, let's talk about HOPE Duchenne. Capricor is a public company. This is a forward-looking statement. That means I might make forward-looking statements. You can find printed copies of our statements on the SEC website, and thank you for paying attention to that information. So I think we've already talked about who's on the call today, and so I'm going to go forward and talk to you a little bit about the data. I think all of you know what Duchenne muscular dystrophy is, but what you may not realize is that the muscles are trying to repair themselves from the minute they start breaking down. So that's why the younger children with Duchenne have the ability to sustain muscle function. And over time, what happens is as the muscle loses its ability to regenerate itself, so then the muscle fails, and the young man serially over time loses function. What we are looking to do with our therapeutic is to help reverse that process by calming inflammation and then by ultimately allowing natural repair processes to occur. We hope to re-stimulate growth of healthy muscle and allow function to either return or at least attenuate the decline. The preclinical studies we've shown in webinars with PPMD before and with Coalition Duchenne as well, where we have shown that in the MDX mouse, which is a genetic model of muscular dystrophy, and one of the ways we can test therapies for people with this disease, the mice had improvements in their cardiac performance in terms of ejection fraction, which is the ability of the heart to meet the needs of the body, and also in the amount of scar in the hearts of these mice. What we also noticed is that the mice were able to run farther and longer on a treadmill, and this suggested perhaps that there were effects on skeletal muscle as well as on cardiac muscle. And we were able to confirm that by taking out skeletal muscle from the mice and testing them and making sure that they in fact did work better. So this then led to the design of the HOPE trial, which I'll explain in a moment. CAP-1002 has been in 3 clinical trials looking at scar in the heart: Caduceus, All-Star Phase 1, and now All-Star Phase 2, which is trials looking at patients, adults with heart attacks, and then in Dynamic, patients that have advanced heart disease. So these cells have been in clinical trials for a while. They're a progenitor cell, which means they're not really fully a stem cell. They can become certain cells, but they can't become all cells. And what they do is they function as a local drug delivery system where they go in and they deliver a payload of contents called exosomes that allow the cells to mediate their beneficial action of anti-fibrosis, reducing scar, anti-apoptosis, which is preventing cell death, pro-angiogenesis, making new blood vessels, and anti-inflammatory, which means that they calm down the inflammation in the area. If you're interested in reading about the preclinical work that's been done starting in 2011, as Cap just said, so it's a magnum opus, if you will, of preclinical work to get us to this point today. It is shown in this website on bioRxiv, where you can read the academic paper written by and published by Eduardo Marban, the clinical scientist who has worked so hard to get this therapeutic to the point that it is today. So shown in this slide is a diagram, or a cartoon as we call it, showing how we expect the cells to mediate what we call their mechanism of action, so how they mediate their beneficial effect. So the cells are delivered. In this trial, they were delivered via a coronary blood circulation, all 3 coronary arteries, which we'll talk about in a moment when I show you the clinical trial slide. But imagine the cells are getting into the blood, and when they get where they're into the blood, they release a lot of these little tiny particles called exosomes. And exosomes are nanometer size, which means they're 1/1,000,000th of a millimeter, which is extremely small. And they have contents inside that basically tell cells what to do next. So it's called epigenetic modification, and what that means is that you can take the cells and tell it to behave differently by making different proteins. And by telling the muscle cells that are already in the body to do different things, we can then target the endothelial cells, the fibroblasts, the myocytes, and even the macrophages and the white blood cells, which is how they modify inflammation. We can actually isolate these exosomes from our CDCs, and we can recapitulate the mechanism of action of the cells. So we are knowing for sure that the cells behave by releasing exosomes. You can also block exosome production in the cells, and you can block the activity of the cells. Now, Eduardo has done a lot of work where he's actually isolated contents from the exosome, so we even know what goes inside the cell and what makes the difference. But for the sake of brevity, we're not going to be talking about that today. So what we're talking about today is the HOPE Duchenne clinical trial. HOPE Duchenne was the brainchild that was developed by Pat, Kath, many other thought leaders, John Jeffries, our PI, came together and designed a trial that we thought could potentially test the power of these cells in treating Duchenne muscular dystrophy. When we began this trial, we had 2 goals in mind. One, we wanted to have a usual care control group. We know how important it is to be in a control group at the same time so we can actually measure the activity of whatever therapeutic you're testing against people that are basically at the same point in their disease process when they enter the trial. So I want to say a thank you if any of you are on this call that were in the usual care group, or if you know anybody that was. Your service to the community was really, truly tremendous and self-abnegating. So very important to have that as to measure against so that we know what we can expect from the therapy as we move forward. The Food and Drug Administration mandated that the first few patients in the trial be 18 years of age or older. This was largely due to the fact that they had to undergo a coronary catheterization. They wanted to make sure that those that selected it for themselves. But then once we got through those first few patients, we were allowed to treat patients that were 12 years old and older. Again, that was because of the size of the heart, and since we were doing coronary catheterization, we wanted to make sure that the hearts were big enough and that we could very easily use adult-sized catheters to put the cells into their coronary vessels. All the patients had to be on steroids so that we could make sure that everybody coming into the trials would have had the same kind of therapy coming into the trial. And they had to have scar in their heart. So that was the most important thing in this trial. We started off with the idea that we were going to be treating primarily the cardiomyopathy, and they had to have a scar in more than 4 segments of their heart, which is about 20% of their heart, plus or minus, so that we could then see if we could reduce that. And also remember, we have almost a decade of experience of working with these cells in people, and so we knew that was sort of a good point at which to begin where we could potentially see changes or improvements. And they had to have ejection fractions, that is the ability of the heart to meet the needs of the body, at a point at which they were still pretty stable in their disease process, because we wanted, again, not to have to deal with patients that might be needing some type of intervention for their heart that was more than just observational over the months of these studies. We worked at 3 sites, extraordinary and amazing sites with amazing physicians. Cedars-Sinai Medical Center under the direction of Ron Victor, Cincinnati Children's with our own John Jeffries, who provides so much service to the community, and University of Florida with Barry Byrne, who I have watched through my career as he has made his way through therapeutics in many types of heart disease over the past 20 years that I've just been paying attention to his work, and I'm so delighted to have the opportunity to work with him. So what we did in this trial was we infused the cells into the coronary vessel. So when we started in this field, and as I mentioned, we've been doing this for a while, we thought that we had to do an infusion into the coronary circulation so that we could get the cells to the heart. And, we believed that the cells getting where they needed to be was of primary importance. And so the patients undergo this catheterization procedure. It's shown diagrammatically and pictorially here. You can see a little vial on the side, a little bag of cells, tiny. They're thawed. It's a frozen product. It's an off-the-shelf product. So what happens is when a patient comes into the trial, or someday when the physicians need to use it, they'll call the pharmacy. It'll be taken out of the freezer, brought down. In this case, it was brought to the cath lab where it was thawed at room temperature, and then safely infused into the coronary vessels in the heart. Now, in the next slide, we'll show, and, there were 12 or 13 guys in this trial that underwent this procedure. This is shown here, a patient being infused in a cath lab. I think I recognize that PI infusing the cells as somebody who's been in one of our adult trials. And so, this is a very easy procedure. The guys that got it, you can ask them. It didn't really cost them too much time. I don't think it was very uncomfortable, and they got through it just fine. Certainly, for a procedure that could make you better over time, it's well worth going through. But as you'll hear at the end today, one of the things that we're most excited about is we can move this potentially out of the cath lab, and our current goal will be to infuse the cells systemically into just a regular, normal blood vessel that won't require going into a procedure room. And so we think this is going to be a big step up. But, a thank you to the guys that went through this in this trial, because they've allowed us to make these observations and has really helped move this field forward. So these are the baseline characteristics of the patients that came into the trial. We knew, because we were targeting 1, older boys and young men, and 2, those that had significant amount of cardiac scars, that we were likely to recruit patients that were further along in the disease process. As you can see here, our thoughts were, in fact, true. We have a significant proportion of the patients. In fact, in total, two-thirds of the patients in the trial were always using wheelchairs, non-ambulant. What you also see, and it happens a lot in small trials, it's typically why those that design clinical trials, like bigger ones, is that even in the luck of the throwing of the die, sometimes there's not exactly equal balance between the groups. And you can see that those that got the therapeutic through no real reason ended up to have more impact in terms of non-ambulance. Nearly 80% of the patients were wheelchair users. Let me just say, at this point, we at Capricor are so delighted that we are able to offer this therapeutic to patients that are farther along in the disease process. We know that many of the clinical trials that are going on now and that have gone on in the near past target younger patients with the hope of attenuating disease. And we certainly think that we'll be there in the future with this therapeutic. But what's really terrific is that we are able to offer something to patients that are farther along the disease process, that are potentially more needy in terms of therapeutics, and also that are really able to see the benefits in a short period of time. So we fondly call you the hope-like patients, and we will continue to develop this therapeutic for these patients, having seen such success in this trial. The patients had approximately 20% scar on their heart. As I mentioned, that was what we were targeting. Again, just by happenstance, the ones in the treated group had slightly less scar coming in, and everybody had a near normal ejection fraction with 49% of being really close to normal of 50 to 60%. We are agnostic to the type of DMD that the patients had, and again, saw some differences there. We don't believe it causes any change in therapeutic efficacy. We'll look at that over time, but none of the top-line data suggests that the type of disease derivation is going to impact downstream consequence of the therapeutic. Shown here on this slide are the things that we were looking for most importantly, which are the safety outcome measures. I think you can see as you scan down, you see a lot of zeros, which makes everybody very happy. It means that nothing really of any significance happened in the trial. You'll notice that there was a cardiac enzyme elevation in all of the treated patients. We'll have Dr. Ashheim talk about that for clarity in a few moments if there's any questions about that. Suffice it to say, this happened as a result of the procedure infusing into the coronary arteries. There was no downstream sequelae for this, no clinically significant events. The DSMB had seen these and didn't raise any level of concern. I'll say, from my perspective, that I'll be very glad not to have to infuse coronary arteries in the future because anytime you see something like this, it's nice to be able to not have to deal with it, but no sequelae, no problems, no issues created, and happy to take any questions on that at the end of the call. Safety and tolerability, again, the patients did very, very well. The only events that were of notice were several of the treated patients ended up with what they call atrial fibrillation. This was procedure-related. It resolved. Again, no sequelae, no problems. Again, DSMB has seen this. Some of the patients had nasopharyngitis. I'm told as a non-physician that that means they had a cold, and actually I don't think that that was specifically attributed to the therapeutic, but since it happened around the time of that, we recorded it. The one thing that we did have is one of the patients in the trial ended up with fever and confusion. This was a patient in the treated group. Although a lot of digging was done as to what was the cause of this, we never really were able to figure it out. We're very grateful that he recovered and is doing very, very well. But, you know, was certainly aware that this occurred with this particular patient during the post-procedural time frame. So let's talk about the exploratory efficacy endpoints. I think I can say in conclusion the trial was safe. There were no events that occurred that were of any concern. The procedure was safe. This procedure is done in cath labs across the world, literally millions of times across the course of days and weeks. It's mostly done in adults that have some type of heart disease, like a heart attack. And so the people who do this job do it all day, every day. And it was safe in terms of the procedure, and it was safe in terms of the outcomes. What we think is most interesting, and what I'm going to spend the rest of my time talking to you about, is the exploratory efficacy endpoints. So, we had the preclinical data, as I mentioned, in the mice. And one of the things that's really terrific, and I'll say this as a sidebar now, is that the data that we collected in the mice seems to mirror almost exactly what we saw in the people, which doesn't happen all that often, and so provides a really terrific roadmap for us going forward as we design the future clinical studies of what we've seen in the mice. We, of course, focused hard on the heart. As you know, we started off thinking that we were going to look to reduce scar and improve function in the heart. We decided that the gold standard is MRI for this. Those of you that have this Duchenne muscular dystrophy who are on this call know that now it's become part of your standard of care to get a cardiac MR. So that physicians can track what's going on in your heart. Because of the dye that's used, we don't like to do this too often. It's not so healthy to give it too often. So they got an MRI at baseline, and then they got an MRI at 6 months. And some of them have already gotten, and the rest will get an MRI at 12 months to look at scar and function of the heart. It is really good to look at function in the heart and scar using MRI. It is the very best. It's not exactly a test that you want to do very often, so we may actually add in some other measures to look at cardiac function in the next trial that's a little less time-consuming and doesn't involve dye. Stay tuned for that as we flesh out this protocol. Then, as I told you, we had seen this really exciting data in the skeletal muscle of the mice, and so we built in some parameters of functional tests of skeletal muscle. We looked at 6-minute walk test for those that were ambulant. I'm really glad that we didn't stick only with that because, as I said, 70% of the total patients and 80 of our treated patients were unable to complete this test. So we only had a very few. So we also, just to hedge our bets, because we thought we might get kids that did not have the ability to walk, we also built in the relatively new but well-validated test called the performance of the upper limb, where we then can test skeletal muscle function in arms and hands, which, is obviously very, very important in maintaining quality of life. And then we also built in a spirometry protocol to see if we could catch, opportunities and improvement in respiratory function. In the mice, we saw these incredibly beautiful responses in diaphragm muscles, which are largely responsible for inspiration. And so we wanted to see if we could catch it. We were very early in our understanding of respiratory physiology and its measurement in Duchenne muscular dystrophy. And so some of the measures that were collected, thankfully non-invasively, were not ones that are specifically good in a neuromuscular disease. So we'll be working hard with our thought leaders to make sure that in the next trial we see if we can catch that a little bit better. We also did some quality of life measurements. We looked at the PEDS-QL and the POD-C adolescent questionnaire, and we looked at biomarkers, which the data we do not have yet for that, but sort of things if we could catch in the blood would be really great for predicting path forward. You can see that the skeletal muscle testing, the spirometry, and the QOL could be given far more frequently than we could do MRIs. So we have some nice data at 6 weeks, 3 months, 6 months, and 12 months. We're incredibly glad that we did that because this data has now defined our path forward. So again, I can never stop thanking the patients enough. Thank you for schlepping back in time and again to go through these tests so that we have this very beautiful, very interpretable data set. So the first thing that we saw, which I think is of the most importance in terms of what Kath talked about in the beginning of reducing scars, we saw really beautiful reduction in the amount of scar in the heart in the patients that got the cells. The patients that got the cells are shown in red. The patients that were in usual care are in the blue. Now, you see a wide variability there. That's not surprising. Measurement in scar and Duchenne hearts is very, very challenging. It requires a trained eye and significant experience because, as you can imagine, the scar in Duchenne hearts is all over the place, and so you have to be trained as to how to know exactly how to circle that up and measure it. So there's a little more variability than we typically see when we measure a scar and, say, a heart attack. But the trends are unmistakable. This is also global scar, and I will tell you that this was something that I wasn't actually expecting to be able to see. I didn't think that in 6 months' time we would be able to see something as important as reduction in global cardiac scar in the treated patients. So we definitely are taking a pause here and saying this is what we believe to be very relevant reduction in scar and something that could be predictive of cardiac performance in future clinical trials. What we also saw, and what I think is even more performance, is real hints of improvement in cardiac function as measured by systolic wall thickening in CAP-1002 treated patients. Now, I needed somebody who has experience in MRI of the heart and other imaging modalities to explain this to me. I'm going to try my best to explain this to you, and thank goodness we have Dr. Jeffries on this call for questions regarding this parameter. Basically, in a normal, healthy heart, the heart ejects blood into the body by sort of wringing it like a washcloth. So there's ejection of blood that is both longitudinal and circumferential, or in my naive way of looking at it, horizontal. So up and down and across. And this sort of squeezing allows blood to be effectively ejected out of the heart and into the body. In the patients with Duchenne muscular dystrophy, even though their ejection fractions seem normal, they develop what is called kind of a restrictive cardiomyopathy, and that's because they seem to have lost the up and down or the longitudinal ability of their hearts to contract and are really left with this horizontal or circumferential ability of the heart to squeeze. So if you kind of just now think of it like squeezing, I think Dr. Lima, our MRI thought leader, said it's like rolling up a pancake and trying to squeeze blood out of sort of a rolled pancake, or I prefer to think of a pancake in syrup, but anyway, think of syrup squeezing out of it. And so what we see here is that we see an improvement, and this is statistically significant in the inferior wall, and then trending beautifully in the anterior and lateral wall, improvement in wall thickening, which wall thickening then means the ability of the heart to squeeze in that horizontal way to eject the blood out. So this is important for multiple reasons. One, wall thickening may be an important indicator of cardiac output. That's the total amount of blood that can be put out of the body in a minute in Duchenne muscular dystrophy compared to, say, a normal heart. What's also important and has really caught our attention is that it's the best in the inferior wall. So in the inferior wall is where the scar aggregates the most, is seen to be the worst, and is predictive of outcomes. So while this still requires study on our part, and we're still waiting to fully look at some of the segmental scar data, if in fact we saw the scar go down in the inferior wall, that is in line with this improvement in function in the inferior wall, it's not unreasonable to say that there could be downstream effects on things like ejection fraction or, going forward, other ways of measuring whether we've improved heart function in a big and more global way. So that is the wall thickening data. Patients came into the trial. They had near normal ejection fractions and volumes. Volumes are the amount of blood in the heart at the end of a beat, and then right before it's ejected, at the end of a beat when the blood is at the lowest, and right before a beat when the blood is at the most in the heart. We use these to sort of quantify how big the heart is, and as a heart gets bigger, it becomes less and less effective at meeting the needs of the body. The hearts in Duchenne muscular dystrophy patients are more similar to a type of heart failure called heart failure with preserved ejection fraction. This is actually well known in older people and is well characterized and actually paradoxically very hard to treat because you don't see these reductions in ejection fraction that we're typically used to seeing in heart failure. So patients with Duchenne have normal or near normal ejection fractions until very, very late in the disease process, and normal or near normal volumes until very late in the disease process. But what I saw when I looked at this data, and I have to say, when I saw these red bars and blue bars myself, I thought, wow, this is moving in the right direction. We're seeing what could be considered a movement towards improvement in ejection fraction and what we could consider a movement towards what we would consider an improvement in volumes. And even though it's near normal, it's still signifying that the cells may be deriving a beneficial effect in global cardiac function. So we're going to watch this carefully over time and in the next trials and see if we can discern whether or not the cells can make a difference in global indicators of cardiac function, such as ejection fraction and volumes. So that concludes, sort of the top line cardiac data. We still have more data that's coming in all the time. I saw some data recently that, from the heart that will further validate and substantiate some of these findings, and I'll look forward to sharing those with you over time. I'd like to now turn our attention to the performance of the upper limb test. So I think those of you that are patients and those of you that are parents of patients have actually seen this test being done. For those of you that haven't, it's a really amazing test that was developed by people, and I'll call out our thought leader, Michelle Eagle, who is at Newcastle in England, and Eugenio Mercuri in Italy, who has been extremely helpful to us in interpreting this data. We have other thought leaders like Dr. Montoni and Dr. Voigt, Dr. Finkel, Dr. Byrne, who is all who have helped us understand the ramifications of this very unique Dr. Victor data set. Some of them tasks that are designed in the poll are to mimic activities of daily living. So we can think about brushing teeth, brushing hair, drinking a can of soda, hugging your mom or dad are some of the things that can be done when you have ability to move your elbow, move your hand, or potentially move your shoulder. As I mentioned earlier, we only had a very few patients that could do the 6-minute walk test, so we are not showing that data because it really doesn't provide any clarity because so few patients were able to participate in that aspect of the trial. Shown here are the tasks of the performance of the upper limb. They're divided into the shoulder, the middle, and the distal. Shoulder level abilities are lost soon after ambulation is lost. The entry test is lifting upper extremity weights. Only a very few of the patients in this trial could do that, and so we didn't really have data that we could use as comparison. And as you'll remember, patients that got CAP-1002 were more impacted in terms of ambulation than those that were in the usual care group, so there was kind of a disparity in the number of patients. We couldn't really draw any conclusions from that. What we were able to do was collect data from every patient or nearly every patient in the middle and distal level. As you may know, the distal level is the last thing that is lost. It's fine motor skills, and so every patient in the trial was able to do that, and most of them were at normal or near normal levels. So we focused hard on the middle level, which actually is also to consider to be very important because maintaining use of your elbow, as I said, allows you to brush your teeth, feed yourself, hug your mom. Note I say that multiple times. Largely because I'm a mom, and I know how nice that would be. So what we see here is really interesting data, and I want to share not only the improvement in the cell-treated group with you, which is evident, but also I'd like to call your attention to the time course of the effects. So we see very important, I think, improvement in the performance of the upper limb at 6 weeks and 3 months post-treatment with a leveling off by 6 months. This is compared to really no change in the usual care group. Remember, I said the time course was important. So what we see here is the peak of the effect, let's call it somewhere between 6 weeks and 3 months, and dropping off somewhere before 6 months. This is exactly the time course that we saw in the preclinical data, exactly what we saw in the mice. Those of you that participated in thought leader panels or that we had talked to before or have been on previous webinars have heard me say over and over that we don't know what that's going to translate to in humans, and now we have the answer. And it's very exciting for us because now we can plan our redosing based on the data that we've seen. So, look for in the next trial, redosing, 3 months, somewhere between 3 and 6 months. We're still trying to work out those details of how it's going to be done. But, we're laser focusing on that time frame because we see a marked improvement in the performance of the upper limb at the mid and distal levels, otherwise called the pull excluding the shoulder, in the patients that were treated with the cells. We then did a responder analysis. So what we really wanted to figure out is, what is a real improvement? And when we talked to, Dr. Mercuri and Dr. Eagle and, sort of got their impressions of, what would be important here, they said an improvement of 1 to 2 absolute points in the scale would be considered, clinically important and relevant. And so we decided to go a little bit further and say, okay, in order to be considered a responder in this trial, you have to have had an improvement of around 3 points on an absolute scale. And now you see even more substantially the improvement in the treated patients. The treated patients had a statistically significant improvement at 6 weeks. Significance had dropped off by 3 months, but was still very evident in terms of improvement. And even at 6 months, you're still seeing a somewhat improved picture in the patients that got the cells. And the level of improvement was really quite good with most of the patients at 20% or better. So they had a real significant improvement in the mid and distal performance of the upper limb in this responder analysis. And finally, I'd like to show you something that I think is the most important piece of data in this clinical trial. So what we did is we said, okay, we have patients that have improvements in scar and function in their heart, and we have patients that have improvement in the performance of the upper limb. Do they go together? Are the same patients responding? And so we did a dual responder analysis, and what we saw, and you see in the graph on the left, and remember red is always treated and blue is always usual care, you see in the graph on the left that when we looked at wall thickening, the measure of cardiac performance, and we compared that to patients that had improvements in the middle level pull, we see a very relevant, statistically significant number of patients that were dual responders in the treated group compared to those that were in the usual care. And then we did the same analysis in scar reduction and the middle level pull. And again, same basic results. We see that those that had an improvement in their heart also had improvement in the skeletal muscle. So what that says to us is that the cells are working all over the body and that they are in fact, not seeing improvement in heart performance or skeletal muscle performance by chance. This is something that is largely and most likely due to a treatment effect. So finally, I'll close with showing the little bit of data from the PODC, which is a quality of life indicator. As those of you that take these tests know, they're sort of looking at how happy you are, pain and comfort, activities of daily living. We see a statistically significant difference at month 3. It's intriguing to us because that is definitely when we were seeing the best performance in terms of the muscle measures in the treated patients. You'll note that the significance is largely due to the usual care group actually being less satisfied with their life on this adolescent questionnaire than, in fact, an improvement in the treated group. What this says to us, and actually our thought leaders have talked to us a fair bit about this, and Pat's been really helpful in helping us understand this, is that this potentially is a very sensitive measure of the state of mind of the patients. That, maybe by month 3, those in the usual care group were feeling down and out for whatever reason. And so if it's sensitive, we can potentially use it in the next clinical trials. This is a very small trial, 25 patients, started off to look for safety and tolerability. We're really excited to move into the next set of clinical trials, and we definitely will plan on using some of these quality of life measures to see if, in fact, over time, the patients that get the cells start to really feel better as well as function better. So let me just conclude my portion of this webinar by saying that there were statistically significant improvements in exploratory efficacy measures of cardiac and upper limb function in the patients who got the cells, CAP-1002. The clinical results are in line with the preclinical data that illustrate the bioactivity of CAP-1002 on both cardiac and skeletal muscle in Duchenne. Very, very exciting and important because it allows us to hark back to our preclinical model as we build out future clinical development programs. And the drug was definitely generally safe and well tolerated by the patients in the trial. And then finally, I'd like to talk to you a little bit about exploratory efficacy benefits we're seeing, and the limited duration of effect following a single dose shows that to sustain benefit will require repeat administration, and that will definitely be part of the next trial design. So what are the next steps? We have requested a Food and Drug Administration meeting to review the HOPE data and discuss the development towards potential product registration in Duchenne. We're planning to submit the HOPE data to the FDA in support of something called breakthrough therapy, or for cell therapy, there's something new called the RMAT, Regenerative and Medicine Advanced Therapies. That's part of the 21st Century Cures Act, and basically it allows us to get in a shorter line to be reviewed by the FDA for cell therapy and to help us get this therapy through the approval process, and gives us, people that are used to reviewing cell therapy so that we have the greatest chance of getting this therapy out to the community at large in a shorter period of time. The next trial will likely be infused this way. We have to be very careful to say that, we have to take this to the Food and Drug Administration. We have to get them to think that this is relevant and that the preclinical data that we have to support systemic delivery, which is in those same mice and shows the same beneficial effect as the heart delivery, meets their standards. But, we wanted to show you this picture today of what it looks like to get a systemic infusion as opposed to, if you remember, back to the cath lab infusion. So both are safe. Both, lead you to potentially the same outcome of going home and going about your business. But, I think one would agree that this little boy with his tiger looks a little bit more comfortable than having to lay down and go through a cath lab procedure. We will continue to explore our HOPE-like patients, which will be agnostic to ambulatory status or genotype. We will look towards this systemic intravenous delivery, which allows us not only to maintain, the comfort of the patients, but also allows us to infuse more cells and also more often. And we'll be targeting skeletal and cardiac muscle function, and our preclinical data suggests that this should be something that is very doable. Let's hope that the FDA agrees with us. And I guess it's up to me now to say thank you, thank you, thank you. I cannot say enough how important your actions as a community, Parent Project of Muscular Dystrophy, Cure Duchenne, Coalition Duchenne, the JET Foundation, are to moving therapy forward for boys and young men, your sons and brothers and husbands with Duchenne muscular dystrophy. It is with grassroots efforts like this that we actually can make the world a better place. And so it is my personal honor to work with all of you. Thank you to the patients, and I sincerely hope that our drug continues to bear fruit in terms of clinical efficacy so we can actually do something for this really, really debilitating and ultimately fatal disease. So I'll thank you for your time. I think that's the end of me, and I'll turn it over to who's next.
This is Kathy. Thank you so much, Linda. That was a great presentation, and I think everybody really got their questions answered, and you were able to shed some light on what has been a really important trial in this space. We all recognize that the real heroes of this trial are the boys and their families that endure everything that is required to push future research and novel therapies forward. Dr. Jeffries, did you have something that you wanted to add as well?
Yes, I think, Linda, thanks again, a wonderful job. And I think some of these data, as you can appreciate, we see these divergence in where we're seeing the numbers going the right way in the treated groups. As everyone probably appreciates, it's a pretty small number of patients. And one might be able to think that if we had more patients that were treated, that obviously we might see a bigger difference in the positive direction. I think the other exciting aspect is the idea that we can potentially avoid coronary infusion, and the systemic infusion is a possible delivery route, one that makes it so much easier a lot more applicable when it comes to patients who can come into an outpatient setting and deliver this therapy in a very easy and repeated way, I think is fantastic. And the fact that we were doing coronary injection in these patients and seeing potential impact on skeletal muscle function, I think is hugely exciting. And it really begs the question, if we do deliver this systemically, what can we do to the heart? What can we do to the diaphragm? What can we do to the skeletal muscle? I think this is just a fantastic lead-in to what's really going to be an impactful therapy over time.
Thank you so much. One of the questions that I was given the correlation between the POL and the cardiac results. Do you think that there was purely a cardiac effect that then impacted the POL results, or do you feel that there was skeletal muscle as well as cardiac muscle that both influenced those results? Does that make sense?
Yeah, it makes a lot of sense. In fact, this is Linda, obviously, Linda Marban. This is exactly the question that I asked when I saw the preclinical data of the ejection fraction in the mice and then the skeletal muscle function in the mice. And because it seemed to make sense, right, that you would have better perfusion of muscles and therefore you'd have better skeletal muscle function. We can't, obviously, and we're not going to, take out skeletal muscle of human beings to corroborate the fact that it's a direct skeletal muscle benefit, but what I can say is in the animals, we were able to parse it down and determine that they are separate effects, so that the heart is one set of effects and the skeletal muscle is another set of effects. And in fact, what they've seen preclinically over and over, which is so fascinating, is that the skeletal muscle benefit is better than the cardiac benefit. And they're able to see that, and you'll see there's that paper that I showed the link to a little bit earlier, which is also available in other places, so we can make sure you get it, that suggests that the skeletal muscle benefit is really quite profound. And so at this point, we're going to say we think that they're separate based on the animal data. In human beings, it's a little bit harder to parse out. I will say that the people in the trial, as you know, had near normal ejection fractions, and Dr. Jeffries and Dr. Ashheim, both cardiologists, are more capable of commenting on this than I am. So, John, I hear you. So why don't you expound on that a little bit?
Yeah, I don't, the changes we were seeing in thickening per se probably wouldn't account for changes in skeletal muscle function. So I think Linda's point about this being two separate effects is absolutely on target. But I do think this idea of cardiac thickening is a very important idea that really implies that we're having some local effects maybe on a fibrotic level, that we're having more appropriate cardiomyocyte contractility, which I think is hugely exciting. And it's one of those things, once again, if we have the opportunity to do repeated infusions, whether that's systemic or otherwise, the opportunity to see global contractility increase could be huge. And the area, obviously, that we're seeing that improvement in thickening, as everyone knows, is an area that's characteristically involved in Duchenne. So it really gives us an idea that that area that's probably most typically affected may be the one that's starting to see some benefit, which would give us an opportunity to get more and more therapies potentially in the future and see more global improvement in overall thickening and see changes in systolic volumes and other things, as Linda was showing you. And then, Deborah, do you want to expand on that?
I actually think you've covered it nicely, unless there are further questions about it.
In order to see the combined impact of repeated infusions, have you decided whether the 13 people in the HOPE-like trial will be included in this next phase?
So this is Linda. It's really interesting because when Pat actually and Cass, both involved in this call today, saw the mouse data. And what we see in the animal studies is that the second dose actually leads to an even stronger effect, at least in the mice, than the first dose. They started calling me saying, can we start repeat dosing, in these patients right away? And we really bandied about that idea and went almost all the way to the door of doing it. But what we realized is that because patients came into the trial obviously in a staggered way, we couldn't, in a very cohesive way, deliver the cells over time in a repeat dosing fashion the way that we could and really be able to draw conclusions regarding that. So I don't know, Deborah, you can answer whether or not the 13 patients that were in HOPE could come back in for systemic infusion trial, but at least not as part of HOPE, right?
Correct. So the issue with redosing, for instance, in the HOPE trial is that the period of time, in order to be able to complete the 6-month and the 12-month testing to be able to understand the results of first this HOPE trial, that's now looking as if, first we needed to understand what the duration of effect was for the patients who were initially dosed with Duchenne. So since it's looking like we're going to need to do redosing more frequently, that's why we couldn't roll it in to the HOPE trial. We needed to understand that timing first. And whether or not the HOPE patients in specific will be able to then participate in the future trial is definitely one of the things we're going to be discussing with the FDA, absolutely.
Okay, that's great. Thank you so much. One of our questions is, will the future trial have a lower ejection fraction requirement than the original trial? Or have you considered that yet?
Yeah.
Linda, do you want me to run with that?
Yes, please.
Yeah.
Okay, sure. So in terms of a requirement for a lower ejection fraction, first of all, I should back up for a second and say that we are in the process of working with Dr. Jeffries and a number of our other, certainly our current cardiology principal investigators, as well as a bunch of the neurologists who you all know well in the Duchenne space and pulmonologists in order to really refine as best as possible the patient population for the next trial. Obviously, you have to balance the desire to be as inclusive as possible, knowing that, there's certainly a tremendous need within the group of patients that has Duchenne for these investigations, and also the need to make sure that the trial that we designed, the group of patients that we aim to target, are the most precisely honed, chosen in order to be able to understand as quickly as possible what the treatment effect might be, so that we can determine that effect and hopefully get a therapy to approval as fast as possible. So that's a long way of saying we don't yet know exactly what those eligibility criteria are. We will want to be able to, in addition to obviously seeing whether or not there's an effect on the skeletal muscle, so part of what we need to make sure, as Linda mentioned earlier in the talk, in how we refined the patient population for the HOPE trial in terms of the patients that we were looking for to participate, we clearly need to find patients who are similar in that measures like the pool will be as important, and as sensitive, relevant to that group of patients as possible. And also that we will be able to see an effect on the heart as well. So it's not that we'd be looking for necessarily requiring a lower ejection fraction. But we certainly, again, are discussing whether or not we are even going to require eligibility criteria that define the extent of heart disease at all. So again, super early, except that we are finalizing this hopefully in the next couple of months, because again, we're going to be meeting with the FDA in a couple of months.
Okay, so the short answer is that those parameters haven't actually been clearly defined at this point, right?
That's true, yes.
Okay, great, thanks. Were there differences in the baseline cardiac medications of the boys that were on the HOPE-like trial, or were they all on the same medication? Do you think medications had any impact in the results that you gathered?
Deborah?
So great question. There were some differences in the cardiac medications. There was fairly typical use of some of them, ACE inhibitors, beta blockers. But given the fact that, the ejection fractions, as Linda had mentioned, were near normal, not all of them were on cardiac medications. It's really a small population, so, in the trial, so it's very hard. In fact, I would venture to say impossible, given the size of the trial, to really be able to understand the impact of the medications, meaningfully understand the impact of the medications on any of the parameters we were measuring. But it doesn't look as if there was an impact.
Agreed.
Right, and I think we can go ahead, John.
Yeah, no, I think that's right. Many of the patients wouldn't have been on remodeling therapy. Some of the folks may have been on some sort of antifibrotic therapy, whether that was spironolactone or otherwise, but it would be very hard to parse out just because it's such a small cohort of patients. And many of those, if they were on meds, had been on stable therapy.
Right, and I think it's also important to say, just as a follow-on to that, that if standard regimen cardiac medications worked, we probably wouldn't be having to develop a therapeutic. So I don't think that there's any evidence that one cardiac medication or another would in some way impact the effects of the cells. They work very differently.
While we're on the line of medications, has there been any consideration of combination therapy, such as combining this therapy with exon skipping or any other therapies that might be out there at the moment?
Well, we've been saying since, we started, even before we started the trial, that we're good friends in the sandbox. And so we are open to combination therapy. At this point, we haven't had any formal relationships with any of the other companies developing things like exon skipping, but certainly would be amenable to such.
Okay, thanks. Also, since you've seen a decrease in scar in the myocardium, would you expect to see a decrease in scar in the skeletal muscle as well? And have you considered how you might evaluate the skeletal muscle in the next trial?
Yeah, so the preclinical data definitely suggests that you see a reduction in scar. I mean, it's actually quite profound, and there's some pretty pictures in there. And I would encourage especially the older patients that want to look at that, to just see red in the slides will be fibrosis or scar, and lack of red shows scar going down, and you can see it like black and white, which is great. So we expect that the same thing happens in the patients. We are not sure how or if we're going to try and measure that in the next trial. We might build in some exploratory efficacy endpoints to look for scar and skeletal muscle. As many of you know from having followed this field for a long time, it's really, really tough to catch that in the biopsy. And it seems to be tough to catch those kinds of things even in MR. So we're looking at those as opportunities. We're certainly open to building them in. We think that that's what's happening, but what we're saying now is that since function is improved, we're happy with that for right now. Deborah, I don't know if you want to add anything to that if you want to, but if I missed anything.
No, I think you haven't.
Okay. All right, excellent. I think we have answered the questions that I've received, and I think we're just about out of time. Cass, I wondered if you had some closing remarks?
I just want to thank you so much to, firstly, the boys who are on the trial. That was an incredible. I know that Dusty's been on trials in the past. It's a huge commitment and offers so much for the community. And also to Dr. Eduardo Marban, just amazing scientist. And, of course, the Capricor team. You, Linda, have done so much. John Jeffries, thank you so much, and Deborah as well. I'm just interested to know with the next stage would be systemic intravenous delivery. Can you tell us when, you hope that would be? Will it be in a few months? Will it be next year?
It's as fast as we possibly can go. My team will tell you that I have been relentless and will continue to be such in driving this as fast as we can, and our goal is to be in patients this year.
Oh, wonderful. All right. Well, thank you so much. Thank you.
Thank you.
Thank you. Thank you, Kath. Well, this has really been extremely interesting and really informative, and we are so excited to partner with you and hope that we can help your work move forward. So thank you all very much for joining us today. Thank you very much to our presenters, and this concludes our webinar.